Hello. Now I am going to tell the interaction between the host and coronavirus. Coronavirus and host. How it destroys host cell and microorganism action, how it infects, and how actually the virus infects inside the cell called virulence, and how it damages the host cell. This is the structure of coronavirus. This is how it looks. It does not have a machinery of its own. It has protein coat, and this is most dangerous protein called S protein. S protein or spike protein. It's a globule, and there is an RNA inside. It does not contain Golgi apparatus, mitochondria, nothing, absolutely only RNA covered by protein wall. Just focus on S protein or spike protein. This is one of the which is important lethal component of virus. This particularly virus going to infect our lungs. Normally, when our season changes from summer to winter or winter to summer or rainy season, there are new viruses which come in the air, infects the enters into the mouth and infects the pharynx. Maximum it is restricted to throat and pharynx, but in case of corona, the virus infects the lowest or innermost part of the lungs. That is why it is dangerous. Now I will tell where exactly the virus acts on the lungs. These are the lungs: bronchi, bronchial branches, and bronchioles. I have magnified this one bronchiole, and this is the epithelium with cilia, mucosal cells with ciliated cells. These are present inside the bronchiole. These cells are the target cells for coronavirus. We know that mucus traps anything which is foreign, which is not required by the body, and cilia drives back into the throat and coughed outside. That's why we get cough. Actually, cough is a protective mechanism. So mucosal cells and the ciliated cells form an important defense line for the any foreign body entered into the lungs. Now the coronavirus enters into the mouth, throat, and lungs and reaches the bronchioles and the mucosal cells. Consider this as a mucosal cell. With the cell membrane. This is the virus. Come in contact with this mucosal membrane. Now how the virus gets attached and entered into the host cell or the mucosal cell. There are certain receptors present there are numerous receptors present on the cell membrane. These are called ACE2 receptors or angiotensin converting enzyme 2 receptors. These are the receptors present on the mucosal cell membrane. Now the virus, the S part of the virus or the spike protein part of the virus exactly fits into the ACE2 receptor. And by endocytosis they enter into the mucosal cell. Now the virus has come inside the cell. Now it releases the RNA in the mucosal cell. In the mucosal cells there are endoplasmic reticulum, ribosome, and above the endoplasmic reticulum there are ribosomes present. Then RNA enters into the ribosomes and the ribosomes start synthesizing the new protein molecules. These new protein molecules are formed and the RNA released enters into and fits into the newly formed protein molecules, and the fresh coronavirus is formed inside the cell. This coronavirus multiplies and many multiplies, and by exocytosis enters, comes out of the mucosal cell and again infects the another mucosal cell. Thus, like this, it multiplies, and there is how new coronavirus or viruses are found, produced. This continuously destroys the lung tissue present in the lung tissues. Thus the and thus this coronavirus penetrates, multiplies, and breaks. For its survival, it uses mucosal cells and ciliated cells, and slow death of lung cells starts, and this is called as apoptosis. Next problem is the body immune system starts responding to this destruction of lung tissue. The immune system thinks that new thing is happening in lungs. WBCs start attacking the lungs. WBCs wants to attack the viruses, but WBC's attack is not specific, not target oriented. It's not like a gun, it's like a rocket launcher, attacks the normal lung tissue along with the viruses and destroys the normal lung tissue also. Because of constant destruction of lung tissue, the debris are formed inside the lungs. Debris are formed inside the lungs due to the continued destruction of the lung tissue. These debris causes choking of the lungs, leading to breathlessness, and at this leading to severe problems. The patient will be put on ventilator, but lung was destructed or destroyed already, so it becomes life-threatening. For attachment of virus to ACE2 receptors is specifically facilitated by an enzyme called TMPSS2 enzyme. This TMPSS2 enzyme is transmembrane protease serine 2 in membrane enzyme, and this is essential for the attachment of and the basis for the attachment of coronavirus to the ACE2 receptors. Now, if you treat the patient, or there is no treatment still for this coronavirus infection, but if you inactive this enzyme, or if you make inactive in this enzyme, we can treat the corona infection very easily, because without this enzyme the coronavirus does not attach to ACE2 receptors, and thus we can treat easily. And the research has to be done in this, and hope in future as early as possible we will success in this aspect. This TMPSS2 enzyme is specifically produced by the from the gene 21 pair of chromosomes. The code will be present. This code will be present on gene 21 pair of chromosomes. So if you make this enzyme inactive, then there will be no attachment, and there is no entry of coronavirus into the mucosal cell, and the lungs can be saved, and there is no further damage to lung cells. So we can easily treat the coronavirus if you find this treatment. This is how the whole pathogenesis occurs. It destroys the coronavirus destroys the lung cells, the mucosal and ciliated cells, bronchioles, and the innermost and Innermost part of the lungs are destroyed, hence it is more dangerous respiratory tract infection. Thank you.